Energy from photosynthesis originates from the sun, and it arrives on the earth as the sun's waves in two forms, both in wave form and in particle form, called the protons. The protons are the particles and the units of light. The protons oscillate along a path which is measured as a wavelength. The light emitted from the sun contains protons in a wide variety of a spectrum of wavelengths called electromagnetic spectrum. Organisms use only a small portion of this electromagnetic spectrum called the visible light. Photosynthetic organisms get their light as streaming protons from the sun. Because they contain a pigment that facilitates the capture of these protons, this pigment reflects some of the protons. This pigment is called chlorophyll. They reflect yellow and green wavelengths, and they absorb red and blue wavelengths. These pigments that are absorbed provide the energy that is used for photosynthesis. Photosynthesizing organisms, which are also known as autotrophs, engage in this chemical reaction called photosynthesis, which occurs within a plant cell. These plant cells are specialized structures that contain the chloroplast. Light-dependent reaction and the Calvin cycle within the chloroplast are small disc-like structures called the thylakoids, which are surrounded by a fluid. It is a space that has got a fluid, and the name of that space is called a stroma. Now the white line here shows the membrane of the thylakoid. The reaction of synthesizing glucose occurs within the thylakoid, in fact on the membrane of the thylakoid. That is the light-dependent reaction, while the assimilation of carbon dioxide or the Calvin cycle occurs in the stroma. Now on the membrane of the thylakoid, there are a couple of structures that I want to introduce you to. Two structures, the green ones, are called the photosystem 1 and photosystem 2 centers. Note here, the one on the left is where the process begins, and yet it is called 2, and photosystem 1 is another site where the system continues. I also want to introduce the BF6 complex. The function that BF6 does, the opposite of it, is done by the structure on the far right. It's a synthase. The small purplish balls are the proteins. They are responsible for transporting electrons, the electric transfer chain. Now let's look at this again. That is the stroma on the outside there, where we'll come to later. That is where the Calvin cycle occurs. That's photosystem 1, photosystem 2 cycle. But on the wall here, what happens is that the protons from the sun come in, and these small balls that we talk about trap those protons because they've got the chlorophyll, and these small balls are called chlorophyll molecules. So chlorophyll molecules capture the protons from the sun. The more protons they capture, the more the electrons within the chlorophyll molecules, the more the electrons will get excited. Once the electrons get excited enough, electrons can be passed through to the outside of the photosystem 2. Otherwise, these electrons will never leave photosystem 2 unless they are transferred through the reaction center. So the reaction center is important as the transfer point of the electrons that have been excited within the chlorophyll molecules. Now, once the reaction center transfers those electrons, it transfers them onto the chain from one proton to the next until they get to the next stage. and now photosystem 1 and 2 receive these protons simultaneously. So although we will look at them separately, but in essence these things occur at the same time. Now remember what I told you about electrons being excited. Some of these electrons can be lost, however they may not all the time make it. Some that are lost, they get lost and they pass through to the inside of the thylakoid. In the inside of the thylakoid you've got water, which is what you see there, the red ball with two blue balls on either side. Now it is the breaking down of that water molecule that will release other electrons, such that the protons that are lost are still useful in sparking another reaction within the thylakoid. So those protons from the sun that are lost spark a reaction that releases electrons. These electrons are then received back into the photosystem. Now when they get into the next site, in essence there, that next site is also making part of the chain. This site, this BF6, actually uses some of the energy from the electrons to pass through the ions. It is important for the ions to be passed through in order to create a gradient of high hydrogen ions within the thylakoid and low hydrogen ions outside the thylakoid in the stroma, so that later on the same process, or the inverse rather of the process, can occur through that synthase, pass through BF6 complex and triggering the passing through of hydrogen ions that create that gradient. But the electrons themselves, weak as they are, get transferred into another protein complex to go to the next site. So remember what I said, electrons get passed into the protein so that they can go to the next site, where hydrogen ions will then be triggered to move through from the stroma into the thylakoid space, and then so that later on those ions can pass through a particular synthase. Voila, it's the ATP synthase. So it's not called by the name of what it transforms, but it's called by the name of what it transforms ADP into. I hope you get that. I hope it helps. ADP, ATP, but it's not called ADP synthase, but instead it's called ATP synthase. Okay, so that's why we need that gradient of ions. But now we were saying that our electrons were having low energy, moving from being utilized by BF6 complex as a source of energy to transfer hydrogen ions from the stroma into the thylakoid space. Now our electrons are coming in. Now they get into photosystem 1. Now photosystem 1 has got a sole purpose of recharging these electrons. That is the sole purpose of photosystem 1, is to just recharge the electrons, give them more energy so that they can be able to execute the same task. But the same process applies in photosystem 2 as it applied in photosystem 1. You need that chlorophyll reactor you need that special chlorophyll for the electrons, for the high energy electrons, to be passed on without losing their energy. They get passed on into the next component of the chain, which is our protein complex, which passes them through to another agent of ours. Now this second agent is different from the previous agent. This agent is having a rather different purpose. This agent is our NADP reductase. So NADP reductase is another agent that is important in using the energy within the electrons. Now pretty much like before, here there is a trick as well. Before, the synthase was called by the name of what it was creating, but this time this reductase is going to be called by the name of what it forms. It forms NADPH, so it's called NADPH ADP reductase. So that in a nutshell is what happens in order for us to get the two important energy forms that are going to be used during the Calvin cycle. That is the two ways in which we get energy transformed into chemical energy. Now, I've got a few foxy questions for you here. What are the two ways in which energy or electron molecules can be re-energized? What are the two ways in which electrons can be re-energized? Now remember, it's not energized, it's re-energized. In fact, to spice up things, give the only way in which electrons can be charged. Clue for the first time. So look at the process again to remind yourself. Try to explain to yourself what happens. Remember what we said, the reasons why all of this is happening, just to give us our two main sources of energy, NADPH and ATP. When these two get outside in the stroma, then we are about to start the Calvin cycle. These need to be supplied in abundance because they get used a lot during the Calvin cycle. Calvin cycle then, like we said, happens in the stroma. Now, the Calvin cycle uses NADPH, and NADPH is involved in this process because the phosphorus in it is needed. Okay, or it's needed to pick up a phosphorus. Its ability to bond with phosphorus that is making it present here. And in the process, the Calvin cycle proceeds. And so both of these really here, if you remember, when they were formed before, it was simply reductions that were made, reductions of phosphorus and adding in energy. So now it is these low phosphate elements, if you may, that are needed here. Now in the Calvin cycle, we're basically dealing with the taking up of carbon dioxide. The process in which we take up carbon dioxide is called carbon fixation. The second process, where there is a reduction process in carbon dioxide, we call that the reduction phase. So it's CR, we fix it, we reduce it, and after that we regenerate. Now I want you to look at this. There are two parts in which ADP can be regenerated along the Calvin cycle, whereas NADPH can only be regenerated once, the rubiscose. Now this part is very complicated, but try to stick with me here. This process actually starts with rubisco. Now rubisco or ribulose 1,5 biphosphate is the starting process here, which carbon molecule that splits into 2,3 carbon molecules. The second step is the sequence of reductions using carbon molecule that splits into 2,3 carbon molecules. Now the main purpose of rubisco here is to capture carbon dioxide. So this process starts with the presence of rubisco. Rubisco captures it as high affinity for carbon dioxide. Now the second phase is the phase that involves ADPH. It's called the reduction phase, where phosphorus is reduced, and another phase is another reduction of phosphorus again. This process is explained again. Look at that. That is a reduction process. Reduction process. Okay. From that reduction process, we get oxygen given off, and we proceed with the glyceraldehyde triphosphate. Now we are approaching now the regeneration of rubisco. There, rubisco is regenerated again, and the process starts again. So rubisco will continuously run around, going to pick up, or rather to deposit some phosphorus molecules. You can form a glucose phosphate, but that glucose can lose its phosphorus molecule, and you know, it can end up bonding with the fructose to form sucrose, or sucrose rather. And you know, these are the forms in which plants use their energy. Now eventually it will be starch and lactose, and starch can also be formed from this glucose. Now we just zoomed in. Now let's recap. We said what we're actually using here in the depth of this process. We're zooming out now. We're trying to produce glucose from energy, water, and carbon dioxide through a reaction that needed the sunlight to produce glucose. Plants need this to have their own energy, but also to develop themselves, and they give off oxygen as a byproduct. I hope this helped you a lot.